Have you ever really stopped to think about what your brain is doing while you're asleep? I mean, it feels like we're just offline, right? But it turns out that couldn't be further from the truth. Sleep is this incredibly active period, a critical time when our brain gets to work, sorting, strengthening, and storing the experiences of our day. It's a process that is absolutely essential for memory. So that brings us to the big question. How does the brain take something you just learned today, maybe a new clinical protocol or a patient's complex history, and turn it from a fragile, fleeting thought into something you can recall weeks, months, or even years from now? Well, the answer lies in this complex nightly process of reorganization, a kind of dialogue that happens deep inside the brain, largely while we're fast asleep. All right, here's how we're going to break it all down. First, we'll look at what we call memory's night shift and the big ideas behind it. Then we'll meet the brain's rhythmic trio, the actual brain waves that are doing all the heavy lifting. After that, we'll see how they come together in a beautifully coordinated neural symphony. Then, and this is really cool, we'll get into how scientists are proving causality with light. And finally, we'll wrap up by looking at the evolving picture and where this incredible field is headed next.
Okay, let's start with memory's night shift. For a long time, the how of sleep's benefit to memory was really up for debate. It basically boiled down to two major concepts trying to explain it. So on one side, you've got the synaptic homeostasis hypothesis. The idea here is that during the day, our brain connections, our synapses, just get saturated with information. So sleep's main job is to just globally waken everything, kind of prune things back to make room for new learning the next day. But on the other hand, you have the active systems consolidation model. This argues for a much more elegant, targeted process, an active transfer and reorganization of specific memories. And for this explainer, we're going to focus on this active systems model.
So this active process really involves a dialogue between two key parts of the brain. You can think of the hippocampus as a kind of temporary scratch pad or a USB stick. It's where we initially save new episodic memories. And then you have the neocortex, which is like the brain's long-term hard drive. During sleep, information gets systematically moved from that temporary storage in the hippocampus to the permanent hard drive of the neocortex. This makes the memory stable and, over time, independent of the hippocampus. But how does that transfer actually happen? I mean, what's the mechanism? Well, it's orchestrated by a trio of distinct brain rhythms, each one playing a really critical role. So let's meet the players in this neural orchestra.
First up, we have slow oscillations. Now, these are very low-frequency waves, cycling at about once per second, and they originate in the neocortex during deep sleep. You can think of them as the master conductor. They create these big windows of opportunity for brain-wide communication by cycling between periods of high neuronal activity, we call those up states, and periods of near silence, or down states.
Next in the trio are sleep spindles. These are short, powerful bursts of brain activity, a bit faster, between 10 and 16 hertz. They come from a sort of back and forth between the thalamus and the cortex. And what's really important is that their presence is strongly linked to learning. They're thought to be the thing that actually triggers the cellular changes, the neuroplasticity, that are needed to physically strengthen a memory. And our final player, sharp wave ripples. These things are incredibly fast, high-frequency bursts firing way up at 100 to 250 hertz, and they come straight from the hippocampus. Now, these ripples are believed to be the neural signature of memory replay. It's the literal reactivation at high speed of the very same neuron patterns that fired when you were first learning something. It's the memory itself being played back. So, we've met the individual members of the band, but their real power, the magic, comes from how they all work together in this precisely coordinated symphony.
Right. It's not enough for these brainwaves to just be happening randomly during sleep. They have to occur at the exact right moment in perfect relation to one another. This temporal coordination, it's the absolute key to the whole memory consolidation process. It's all about the timing.
And this visualizes the entire performance perfectly. The slow oscillation acts as the conductor, right? It creates that upstate, that window of opportunity. Then, nestled right inside that window, the hippocampus sends out a sharp wave ripple. That's the memory replay. And it's immediately met by a sleep spindle from the thalamocortical system. This precise nesting is what allows the memory content from the hippocampus to be broadcast effectively to the cortex, where those spindles help to wire it into our long-term networks. It's beautiful. Now, there is a scientific term for this beautiful coordination. It's called cross-frequency coupling. All it really means is that the phase of a slow wave, like our slow oscillation, is controlling the power or the amplitude of a faster wave, like a spindle. Being able to actually measure this coupling has been a huge leap forward in understanding how these rhythms work together to support our memory. So, for a long time, this was all just a really compelling story, right? It was correlational evidence. We saw these rhythms happen and we saw memory get better. But how could we prove that one caused the other? Well, this is where things get really fascinating. The early methods, like physically waking up animals to deprive them of sleep, they were pretty messy. Sure, they worked, but they also induced a ton of stress. And critics rightly pointed out, hey, maybe the memory problems are because of stress hormones flooding the brain, not the lack of sleep itself. We needed a much cleaner, more precise tool. Enter optogenetics. This is a revolutionary technique where scientists can insert light-sensitive channels into very specific neurons. And now, literally with the flip of a light switch, they can turn precise brain circuits on or off with millisecond timing and without causing any stress at all. This gives us the incredible power to directly mess with sleep rhythms and see what happens. And the results are just stunning. For example, when researchers used light to silence the neurons that generate rhythms during REM sleep, boom! Memory consolidation was impaired. But get this, when they artificially induced spindles to happen right during those slow oscillation upstates, they actually enhanced memory consolidation. They made it better. And when they blocked communication between two cortical areas during deep sleep, the ability to remember a specific texture was just gone. This isn't correlation anymore. This is causal proof. So, this ability to finally prove causality has just propelled the field forward at an incredible pace. And our understanding of sleep and memory is evolving faster than ever. So let's take a quick look at what's on the horizon. Remember those two competing theories from the beginning, the global weakening versus the active transfer? Well, it looks like they're starting to merge. New evidence suggests that synaptic weakening isn't some global passive process after all. Instead, it seems to be an active, very targeted process that happens locally. And it might be driven by the very same slow oscillations that are so critical for consolidating memories. So the brain might actually be doing both at the same time. The future here is just so exciting. Researchers are now trying to figure out how sleep helps us pull out the gist of our memories, not just the raw details. The exact role of REM sleep is still a hot topic of debate. We're also learning that every single person has a unique sleep fingerprint of these brainwave patterns, which opens the door for personalized interventions. And at the most microscopic level, scientists are starting to track memory consolidation down to the level of individual engram cells, the very neurons that actually hold a specific memory. Which leaves us with a final, pretty provocative thought. We are learning to read the brain's nightly symphony, and now, with tools like optogenetics and targeted stimulation, we're beginning to learn how to conduct it. So if we can truly orchestrate these rhythms, just think about what that could mean for treating memory disorders, for counteracting age-related cognitive decline, or maybe even for accelerating how we learn new things. The brain's night shift, it seems, is just beginning to give up its secrets.